Okay, this next part has a little bit of complicated stuff. We're going to be talking about the intestinal phase of digestion. I guess we can erase all this. And that's I'm going to lay out kind of what we're going to approach here. So most of this is happening in the duodenum. So if we sketch in the stomach here as we were emptying, most of what we're going to be talking about is happening in this first 12 inches or so of the small intestine, the duodenum. So after the pyloric sphincter, but before we really get into the next part of the intestine, the jejunum. So here we're going to be talking about, in the intestinal phase, we're going to be talking about the signals that the duodenum sends. So the cells here in the duodenum secrete a number of hormones and send a number of signals to other parts of the digestive system and other areas of the body based on what's coming in here from the stomach. So we'll spend a little bit of time on that. We'll also talk about the role of bile, which is going to be coming from the liver and gallbladder, and the enzymes secreted by the pancreas, which come out in the form of zymogens, and how those zymogens get activated and what they do. And then we will also talk about, as part of that really, what's called brush border enzymes. So here in the duodenum, if we took a very close-up look at the wall of the duodenum here, in the mucosa part of the wall, if we looked very closely at the edge there, what we'd find is cells that have this really, really dense border of tiny hair-like projections that increase the surface area of those cells enormously. Those hair-like projections are called the brush border. So that's along here in the inside of the duodenum. There's a number of enzymes in that brush border that are important. And they are actually important based on some of the stuff that happens earlier. So I'm laying this out so you can try to kind of keep all of it in mind at once as we talk about what's going on here. I'll try to organize it in a way that makes it make sense. So let's start by talking about the signals that the duodenum is going to be sending elsewhere. So as the stomach contents empty into the duodenum has to tell the stomach to slow so it has a chance to deal with the contents. Remember, those strongly acidic contents can be hostile. It also needs to send signals to other parts of the body based on what's coming in to help sort of customize how the body is going to respond to this food. So, one thing I do need to do here is indicate two other things that are going to be involved. That has no ink. And that also has no ink. So, coming down here is the gallbladder and the bile duct. So this is coming down from the liver. This is how bile is descending in this area. Also coming in here, if in red here is the pancreas. This is the pancreatic duct. So pancreatic secretions are going to be coming down this pancreatic duct. And both the bile duct and the pancreatic duct enter the duodenum together, right there at that structure called the duodenal papilla. Which is normally closed by the sphincter of Oddi. So that controls the entry of both bile and pancreatic materials here into the duodenum. So that's something you're going to need to understand when we talk about these duodenal signals. So, as acidic chyme enters the duodenum, the various components of that chyme cause cells in the duodenal wall to send various kinds of signals. The acid causes cells in the duodenum to produce a hormone called secretin. So acid makes the cells of the duodenum produce a hormone called secretin. This was actually the first hormone ever discovered. Secretin does a couple of things. One of its functions is to slow the stomach motility. So secretin is one of the signals that tells the stomach, whoa, slow down, don't send me so much stuff. So we put a little bit of acidic chyme in here, and the secretin is a signal saying that's enough for now. But it also triggers bicarbonate release from the pancreas. So, as the acid comes into the duodenum, the secretin tells the pancreas to dump a load of bicarbonate-rich solution into the duodenum. And that bicarbonate will help to neutralize that acid. So the stomach acid gets neutralized by that bicarbonate. One thing you may have noticed is that there in the stomach, when we made the acid, remember we made proton and bicarbonate, and the bicarbonate went back to the blood. That makes the blood more alkaline. Here in the pancreas, we're doing the opposite. We're taking CO2, making it into proton and bicarbonate, but sending the bicarbonate down the duct and the proton back into the blood. So that actually reverses what the stomach did to the blood. The stomach made it more more alkaline by putting the bicarbonate in there. The pancreas, by making bicarbonate, puts acid into the blood. So it comes out the same. Anyway, so that secretin helps to neutralize that acidic chyme and protect the duodenum. Also tells the stomach, whoa, slow down. Alrighty, so that's one signal the duodenum sends. The acid triggers that. The presence of carbohydrates causes the duodenum to secrete two other hormones. One called gastric inhibitory peptide, also known as GIP, and glucagon-like peptide 1, also known as GLP-1. So, GIP and GULP. The presence of carbohydrates in that chyme causes the duodenal cells to produce these two hormones, GIP and GULP. We used to think that gastric inhibitory peptide's primary job was to tell the stomach to slow its emptying even more. That's hence the name. And it does do that a little bit. But more recent research has suggested that these two both have a very similar function, which is to tell the pancreas to start making insulin. So these stimulate insulin production in the pancreas. So now we're talking to the pancreas, to those beta cells in the pancreas, telling them, make insulin. And if you think about it, that makes sense. If here in the duodenum we detect carbohydrates in the chyme, that's a signal that when those carbohydrates get digested and absorbed, that's going to add glucose to the blood. We don't want our blood glucose to go too high, so this is the duodenal way of warning the body, we're about to get a new load of carbohydrates absorbed through the digestive tract. Get some insulin out there so that the muscle and fat cells can start pulling glucose out of the blood so that we don't end up with a gigantic spike of blood glucose as all of this stuff gets absorbed. So this is a signal to prepare for carbohydrates incoming. Alrighty, so that's the second signal. Second and third, I guess, in a way. There's another one. The presence of fats. Really, this next one will come no matter what comes into the duodenum, but fats cause an especially large amount of it. So fats increase production of has the duodenum produce the hormone cholecystokinin. Great name, usually just abbreviated CCK. So cholecystokinin comes from the duodenum in response to fats especially, but really any food coming in will cause some cholecystokinin production. Cholecystokinin has several effects. One of its main ones is it triggers contraction of the gallbladder. So cholecystokinin from the duodenum tells the gallbladder to squeeze, which delivers a load of bile down the bile duct and into the duodenum through that duodenal papilla. So this produces bile into the duodenum. We'll talk in just a moment about what bile does. The CCK also tells the pancreas to release digestive enzymes. That's another one we're going to talk about in a moment. So that cholecystokinin also acts on the pancreas, telling it, send a load of digestive enzymes down in here. Most of the enzymes that will do the rest of the digestion of food here in the small intestine are coming from the pancreas. And it's mostly cholecystokinin that tells the pancreas to send those enzymes in. In addition, CCK also acts on the brain and other areas of the body. It's one of the hormones that's involved in regulating appetite. And what we have learned as people have studied how appetite works in the body is that it is very, very complicated. As the obesity problems became more prevalent in the United States as well as elsewhere in the world, a lot of research has gone into studying why do people eat? What tells them they're hungry? What tells them that they're full? What tells them to stop eating? And what we've found is that it's a complicated mess. But cholecystokinin is one of them. So CCK from the duodenum is one of the things that tells the body, hey, you just got food. You don't need to eat anymore right now. There are other hormones that are involved in that too, and that go the other way. For example, the hormone ghrelin, spelled like this. Ghrelin, G-H-R-E-L-I-N, is a hormone that the stomach produces when it's empty. And that seems to be an appetite stimulant. Ghrelin says, hey, you should get food. So ghrelin, cholecystokinin, a couple of others, leptin from adipose tissue, all of these interact to help us determine when should I eat? When am I hungry? How do I know when I've had enough? Anyway, so that's one of the things cholecystokinin does. So, just to recap that, as this acidic chyme from the stomach starts entering the duodenum, we have several signals. Secretin tells the stomach, whoop, slow down, and tells the pancreas, send me bicarbonate to neutralize the acid. GIP and GLP-1, which are mostly in response to carbohydrates, tell the pancreas, produce insulin because we're about to get carbohydrates absorbed from digestion. CCK, which comes from anything but especially fats, tells the gallbladder, squeeze, send some bile in here, we've got fats to digest, and tells the pancreas, send me digestive enzymes. We're going to need to digest some food here. It's kind of neat that the duodenum really kind of controls this process. Tells the stomach to slow down, brings in enzymes, bicarbonate, bile, makes the right mixture to make digestion work. All right, so that's what the duodenum is doing. All right, let's take just a moment to talk about bile. So, bile was the stuff coming down from the gallbladder. It comes from the liver as well. The liver can produce bile, which will come down the bile duct. but it accumulates in the gallbladder, which then holds a load of it, so that when you have a big load of fat coming into the duodenum, we can squeeze the gallbladder and send in a lot of fat. This is one of the reasons why people with gallbladder disease, where the gallbladder is inflamed or blocked or otherwise not working, can have very bad reactions to meals that contain a lot of fat, because they don't get all the bile they need to digest that fat, which means the fat stays in a form which is harder to absorb and causes digestive disturbance and make you feel terrible. Anyway, so bile is this complex fluid, but it contains some stuff called bile salts. Bile salts are useful because they are amphipathic, if you remember that word. That means part of them is hydrophilic and part is lipophilic. So let's draw that up here as if they were sort of a little capsule that had a lipophilic part and a hydrophilic part. Now, if we imagine that I've got a glob of fat molecules, so we'll have a fat be this purple thing. Actually, just for consistency, let's have the fats be blue. We'll make them blue rectangles. So here's my glob of fat molecules. Those fat molecules, if you put them in water, remember, since they're lipophilic, the water is going to get out from between them, trying to be around other water, which causes the fat molecules to all get clustered together. So now we've got this glob of fat surrounded by water. If my lipases want to work on this, they all have to work here on the outside, meaning there's stuff in the middle that's not going to be digested very quickly. Bile salts can help with that. When I squeeze a load of bile in here, I've now got these things that are hydrophilic and lipophilic, these amphipathic molecules. And what they can do is get in here and split this up into smaller groups by arranging themselves with the side that's lipophilic facing the fats, but the side that's hydrophilic facing out in the water. So they can get in here and make these things more comfortable separating by surrounding them with lipophilic things, so it'll separate my big glob into smaller globs. My big ball of fat gets separated into many smaller balls of fat, each one surrounded by these bile salts with their hydrophilic ends sticking out, like so. By splitting that up, we now increase the surface area of the fat a lot, which means the enzymes have a lot more places to work on that fat and can digest it faster. This process of taking this big glob and splitting it into many smaller globs is called emulsification. If you're familiar with the concept of an emulsifier, it's something that does this in sort of our food processing industry. If you eat peanut butter, you know that you can go to the store and get the natural peanut butter, and when you get that and you open it up, you find that it's separated into sort of solids and a layer of oil on the top. If you're familiar with natural peanut butter, that's kind of what it looks like. And if you want it to be more like peanut butter, you have to stir it up and mix the oil back in. That's because natural peanut butter does not have any emulsifiers added to it, so the fat tends to stick to itself and make an oil layer. If you get Jiffy or Skippy or one of the other more processed peanut butters, those are all smooth and they don't have a separate oil layer because they contain emulsifying agents. That emulsifier makes it easier for that oil to stay in tiny little droplets and then mix itself up with the solids, the more the proteiny part of the peanut butter, and stay in a more of a smooth mixture. So your bile is doing that in your duodenum. It makes the fat split up into tiny droplets and spread it out in the water so it's easier for your digestive enzymes to get to. If you don't do that, you get big globs of fat in here and they don't get digested as well. So that's just a little bit about bile. Okay, let's talk about pancreatic enzymes. So Now we're going to be looking at the enzymes that are being made by the pancreas and coming down here, tend to be put out there in response to cholecystokinin. Pancreatic. I'm going to call them here zymogens. There's a word for you. Notice the O-gen. A zymogen is an enzyme with a safety cap on it. It's an inactive version of an enzyme. Now, the pancreas, think about what's going on with digestion here. We had salivary amylase digesting starch, but it got denatured in the stomach, so it stopped working. We had a lingual lipase and a gastric lipase. Those were working at the acidic pH in the stomach. But now that the acid has been neutralized, those are now at the wrong pH, and they won't work well. Likewise, the pepsin, which was digesting protein here in the stomach, that was only working at a very acidic pH, like 2. Now we move this to the duodenum, we dump in bicarbonate, we bring the pH up to more like 6, and the pepsin stops working. So all of our digestive enzymes are kaput. They're in the wrong pH, or they went through something with the wrong pH. So we need enzymes to do the rest of digestion. We've still got starches, fats, nucleic acids, and proteins all needing to be broken down into absorbable form. That's where the pancreatic enzymes are going to come from. So the pancreas puts out enzymes for everything. Pancreas puts out lipases, and amylases, and nucleases, and peptidases, both endopeptidases and exopeptidases. So we've got all of these enzymes coming from the pancreas, which means they can digest pretty much anything. They can digest all the major kind of food molecules except some of the big carbohydrates, like cellulose. The problem is, the pancreas is made of those molecules. If I made those enzymes in the pancreas in an active form, they would attack the pancreas. They would break it down. The duodenum, because of some of the mucus on the walls, can protect itself somewhat from that, but the pancreas doesn't. So if I have those enzymes in active form made in the pancreas, I'm going to have a problem as the pancreas self-digests. So that's why when the pancreas makes these, it makes them in an inactive zymogen form. So as they come down the pancreatic duct, they've got their safety caps on. So they get dumped in there into the duodenum, and I've got all my inactivated enzymes ready to go. But if they're inactive, they can't digest my food. So how am I going to fix this? How do I get the pancreatic zymogens to become active enzymes? It's kind of a cool story. But in order to tell that story, I need to tell you a little bit more about the brush border. So remember here in the wall of the duodenum, I've got these cells that have these absurdly long extensions. That's the brush border. Now, in that brush border, we have a number of enzymes. A few of them I'm going to tell you about in a little bit. But one I'm going to mention now is called enteropeptidase. Enteropeptidase is one of these brush border enzymes. It's a membrane enzyme embedded in the cell membrane of these cells that are part of the wall of the duodenum here in the membrane in the brush border, with its active sites sticking out into the duodenum. It's a peptidase, meaning it breaks off pieces of protein. And as it turns out, it's actually going to work to break the safety cap off of one of these enzymes. So let's talk about one of these ones in particular. So I've got my pancreatic lipases, amylases, and nucleases, and then I have my pancreatic peptidases. There's one in particular called trypsinogen. Notice the O-gen, this is the zymogen form, the inactive form. But trypsinogen can be activated by enteropeptidase. So when I put trypsinogen in here, it's going to find that enteropeptidase, and the enteropeptidase will cut off the safety cap, making it into the active form, trypsin, which is also an endopeptidase. Trypsin is then going to activate these other enzymes. So it takes my zymogen nucleases and makes them active nucleases. It activates my amylases and my lipases. So just to recap that, we secrete zymogen inactive forms of lipases, amylases, nucleases, and peptidases from the pancreas here into the duodenum. One of the peptidases, trypsinogen, the zymogen form, runs into the enzyme enteropeptidase in the brush border. Enteropeptidase cuts off its safety cap, so it becomes trypsin, the active form of that peptidase. Trypsin then cuts off the safety caps on all of these others. So the other peptidases become active, the nucleases become active, the amylases, the lipases, all of those now get their safety caps cut off by trypsin, and now we've got a bunch of active pancreatic enzymes in here, mixing with the chyme and digesting all of the biomolecules, breaking them down into absorbable forms. We can have problems with this process. If something blocks this duodenal papilla, like say a gallstone gets stuck here, then as the pancreas makes these enzymes, they can't get out. And as it turns out, if you let them sit long enough, sometimes they can spontaneously do this process. So if I make these enzymes but I can't get them out of the pancreas, sometimes they will start to activate, becoming active enzymes, and then they start attacking the pancreas, and I end up with the condition acute pancreatitis, inflammation of the pancreas as it attacks itself, which is a problem, a serious, immediate problem. Anyway, so once I've activated all those pancreatic zymogens, now I've got active enzymes working on all those food molecules, breaking them down. And chemical digestion now really kicks into high gear. So as all of that moves along down the small intestine, out of the duodenum into the jejunum, and so on, all of that stuff will eventually get digested. Actually, a lot of the digestion happens even just within the duodenum, but certainly most of the rest in the jejunum. And then we're going to be absorbing those. We're going to talk about that absorption in a little bit here. However, I do want to mention one thing in particular. We mentioned that these amylases break down starch into maltose, the disaccharide. Maltose is glucose. There are some other disaccharides in our diet, and the problem is disaccharides can't be absorbed. I can only absorb monosaccharides, single sugars, not the doubles form. So if the amylases only break them down into maltose, which is a disaccharide, how can I absorb anything, any carbohydrates? That's going to be the job of a couple of last brush border enzymes. So let me erase this. Now, if we look at the disaccharides that are involved in our diet, there are three that are of importance to us. Maltose, which is glucose and glucose. That's both from starch breakdown, and plus you find maltose in food. Sucrose, which is regular, when you talk about sugar, like table sugar, granulated sugar, the sugar on your donuts or whatever, that's sucrose. That's glucose bound to fructose. The third one is lactose, which is glucose bound to galactose. These are the three disaccharides that are important in our diet most of the time. And we can't absorb any of those in their disaccharide form. We need enzymes to break them down into those separate monosaccharides. Those enzymes are found here in the brush border. You'll have sucrase, which breaks up sucrose, maltase, which breaks up maltose, and lactase, which breaks up lactose. So, as those disaccharides pass through the duodenum, those brush border enzymes will break maltose up into separate glucoses, sucrose into glucose and fructose, and lactose into glucose and galactose. Once they're in the separate monosaccharide form, we can absorb them. Now that leads to an interesting story, which is the story of lactose tolerance. Mammals, like humans, which nurse their children on milk, and milk contains lactose, mammals produce lactase, they have those enzymes in their brush borders, when they're little. But about the time they stop nursing, about weaning time, most mammals stop making lactase. The production of lactase is reduced a lot so that they have very little lactase left in their brush border, which means they can't digest lactose very well anymore, which is normal because they aren't drinking milk anymore. In humans, there is evidence that somewhere around maybe 10,000 years ago, about the same time that we started domesticating animals, certain human populations had a mutation where they continued to make lactase, the lactase persistency mutations. And thus those individuals retained the ability to digest milk even as adults. There's lots of debates as to why that was selected for, but probably the simplest idea is that if you can digest milk, you have a source of food that doesn't involve killing your livestock. So you can keep getting milk much more easily than you can raise and slaughter animals. So those people had a selection advantage. Their children were more likely to survive and carry on that mutation. And we ended up with some populations of humans who could digest lactose throughout their lives. The vast majority of people stopped making lactase. But now you had a few populations which didn't, and those populations spread. At this point, a little less than half of the world's population is lactase persistent, is able to digest lactose as adults. Most people, the majority of people in the world, are lactose intolerant. They do not make lactase, and so they can't digest lactose well. Although what symptoms you get and how well you can or can't digest it varies a lot between individuals. But certain populations are still more likely to be lactose intolerant versus lactose tolerant. But just keep in mind, the normal human condition is lactose intolerance. Lactose tolerance is the mutation. But it's a useful mutation sometimes. All right, anyway. So that's the final breakdown of the carbohydrates. So now we have digested everything. Our endopeptidases have broken proteins into small pieces, and the exopeptidases are breaking them up from the sides. Our amylases have taken starch and broken them into maltose, and then our brush border enzymes have broken our disaccharides into monosaccharides. Nucleases have broken down nucleic acids, lipases have broken down fats, and all this stuff is now in absorbable forms. And now we're here in the duodenum and later in the intestine, ready to absorb these nutrients. So our last part we're going to talk about is how do we absorb these nutrients? Okay, let's talk a little bit more about how these things are absorbed. So we had several major categories of these molecules. We had carbohydrates, which had to be broken down into monosaccharides. Those monosaccharides get absorbed through the intestinal wall by SGLT and GLUT transporters. Kind of like they did in the kidney, in the PCT, if you remember that. We had proteins, which had to be broken down into amino acids. Really, groups of 1 to 3 amino acids can be absorbed. So little groups of amino acids. Those mostly come in on sodium symports. So symports that will transport amino acids and sodium together. Kind of like the SGLT in a way. Nucleic acids had to be broken down into nucleotides, which have a variety of transport proteins for them too. So, the last category is fats, and I'll get to that one. But I just wanted to lay this out so you see it. Now I'm just going to sketch out for a moment a little bit about what the intestinal lining looks like. So, if we imagine, if here's the lumen of the intestine lining is arranged sort of like this. So this is that inner layer called the mucosa. We've got this outer layer of cells. This is that epithelium. The cells that line that are sometimes known as enterocytes. Those are the cells that make up that epithelium. These little divots down here are known as crypts. And these big projections out into the lumen, one of them is called a villus. Together, in plural, it's villi. What these do is increase the surface area of the intestine a lot. Rather than having a smooth lining, you've got this basically fuzzy lining with these tiny little villi. And these villi contain fenestrated capillaries that come up into the villus and then back out. Which means that for whatever it is that I'm absorbing, apparently all of my green markers are dead. For whatever it is that I'm absorbing, and my blue markers. Okay, let's see. Yeah, there we go. If I can get it through the enterocyte, so for example, if it's those amino acids, I've got sodium-based cotransports on here. Once I get it through the enterocytes into this inner part, I can get it picked up and into the fenestrated capillary and into the blood supply. So that works well for those three categories we talked about, when we've got monosaccharides, amino acid groups, and nucleotides. Those are all small enough to fit through fenestrated capillary fenestrations, so I can get them into the blood. Fats are going to be a different story. So let me erase this side of this, and we'll talk a little bit about what happens to fats. So fats get broken down into, remember, monoglycerides and fatty acids. Those used to be a triglyceride, now it's a monoglyceride, glycerol plus one fatty acid, and then two separate fatty acids. And if here's my enterocyte, here's the funny thing that happens. Being lipophilic, these things can all move into the cell. So my monoglyceride and fatty acids can go right through that apical membrane into the enterocyte. But once they're in the enterocyte, they get put back together into a triglyceride. And then those triglycerides get packaged up with other triglycerides into this big structure called a chylomicron. This is a big sphere of fats and protein. So I glom all this stuff up into a chylomicron, which I then export from this cell. So now we can imagine our fat out here got packaged up here into this chylomicron. So there's my chylomicron right there. That chylomicron is too big to fit through the fenestrations in a capillary. It can't get into the blood here. So instead, what happens to my chylomicron? Do I have anything in here? Nope. Okay. I will have lacteals, which are, hopefully you can see that in orange there. A lacteal is a lymphatic capillary. Lymphatic capillaries have really big holes in their sides. Big enough that my chylomicron, instead of going into the fenestrated capillary, can enter the lacteal and travel out of the capillary, out of the intestine, not in the blood, but in the lymph. So my absorbed fats get packaged into chylomicrons, absorbed into the villi, but they don't go into the blood. They go into lymphatic capillaries called lacteals, which then transport those in the lymph through the lymph nodes and return them to the blood, where the rest of the lymph vessels return to the blood. So my fats skip all of that circulation there, which is kind of interesting. These intestinal blood vessels are going to end up going through the hepatic portal system to the liver, which is going to get first crack at everything that's in there. So if we bring in toxins, the liver can have a chance to try to deal with those. But fats skip the liver. They go through the lymphatic system and end up just dumped into the blood as these chylomicrons. Kind of an interesting thing. So, that's absorption, and that's the end of the digestive system.